Yeah, yeah, for sure. Um, and this is the first seminar, the first time I do participate to the RDN seminar series. So I don't know how you guys proceed on regularly, but if you want to jump in at some point, at any point, and ask questions when I'm talking, please don't hesitate. I may not see you, so you may just interrupt me, and that's going to be fine. Um, it's going to be this first time as well that I give this presentation. This is all new stuff. There's almost nothing published out of what I'll present. And so obviously, eventually some questions may arise as I go through. So please don't hesitate to ask any questions. So I'm a professor in biology at the Université de Sherbrooke. I've been here for a little more than 10 years now. I was previously at Rimouski, towards the northeast of Quebec. And I do, well, I always find it difficult to define myself. I would say I'm community ecologist. And originally I was doing mostly theory. I started in forest ecology, and then I moved to theoretical ecology, and I got interested in many topics, but all of them were more or less related to spatial dynamics and trophic interactions at some point. And I'll touch on some of this today. And with time, eventually I started to do more and more data analysis, and now I'm also running a group dedicated to monitor biodiversity changes in Quebec. So it's called Biodiversity Quebec. I won't say much about it today, but if you're having question, I'll be happy to talk more about it. Hopefully one day it's going to be part of a larger Canadian network for biodiversity observation. You may be aware of what's going on elsewhere in Canada. There are discussions to establish a Canadian biodiversity observation network, and so I'm involved in those discussions. So I could start sharing my screen, and as I said, the work that I'll present, it's all new, and it's work that was done in collaboration with several grad students. But the main ones are Alex Fuster-Calvo and Catherine Hébert. Many of the slides the figures that will show up are coming from their thesis, and some of their last chapters are currently we're trying to publish them. And the main point around the ecological network coherence will eventually come in some of the work that Alex has been leading with the collaboration of Catherine. So I'll start just saying a few words about how what was the thought process that got me thinking about network coherence and bring this new concept. I do collaborate with a group of field biologists who study food web dynamics in the Arctic. Um, I go there now on a regular basis, almost every year, to do some field work with them. Even though I'm going there essentially as a theoretical ecologist, so I'm mostly there just to think about the system and exchange with them. But I have the fantastic opportunity to spend a couple of weeks over there and experience what's going on. So Bylot is a small island that is located north of Baffinland in Nunavut. The field station has been established in around 1990. Originally, it was dedicated to monitor reproducing snow geese. At that time in the 90s, the population was just exploding in North America, and Bylot is one of the main sites where they do reproduce. So there's an impressive colony there where there's roughly 25,000 couples that do breed there every summer. It's one of the most important colonies in the Arctic. But then eventually, over time, the research group diversified and started to study more broadly the dynamics of the entire food web. The advantage of doing that in the Arctic is that, well, diversity on the one hand is relatively low, even though there's quite a lot of functional diversity. The total number of groups is not that high. And the other reason is also that, well, the absence of structured vegetation, the fact that it's, all animals are breeding on the ground makes the observation of traffic interactions much easier. And so it's quite straightforward to do trophic ecology, food web ecology in that system. And so, for those that are not aware of the diversity and how it's organized, the basic species at the bottom of the food web are lemmings. So there are two species of lemmings that obviously feed on the different types of vegetation that you do have there. And these are just at the very bottom of the food web. They're the prey for almost everything. You have highly specialized species such as a snowy owl that feed on them. Snowy owl, they don't show up every year. They mostly show up in the years where the lemmings are at high abundance. Then you have the Arctic foxes who are resident on the island, and so we find them both in the summer and the winter. And then you have a fairly diversified set of predator species, such as the Jaegers and the hawks. We also have the gulls that do predate on the different species. The lemmings are part of their diet, but also you have the chicks of the snow goose. And then there are different other species that are also feeding and breeding on the island over there. And so I've been part of that group and we study their dynamics and obviously given the amount of climate change there is going on in the Arctic, we are interested in understanding how this entire food web will react eventually to climate change. And one thing that we find is, and that it's quite evident, is the diversity of the responses that do have those animals to warming climate. So I was saying earlier that it was minus 30 here in Quebec last weekend. Well, some of those animals are adapted to those extreme conditions, such as the Arctic foxes. So this is a very old study that was published in the 50s, where they made an experiment and they cooled down the ambient temperature and they monitored the basal metabolic rate of the different some different vertebrates that do live in the Arctic. So you have foxes, dogs, polar cubs, polar bears, and ground squirrels. And you can see that for the foxes, their basic metabolic rate is simply not reacting at all to a gradient of temperature that goes from minus 30 to plus 30. So they are extremely well adapted to that cold environment, meaning in this case that if even if temperatures do increase, they won't directly react to that change. But some other species are very sensitive. And so the snow goose, for instance, they get there in the spring, and the key variable that is influencing the breeding success of the goose is the date at which the snow melts. And so here you have time series for different years showing that there's a lot of year-to-year variation in the date at which the snow cover essentially disappears. And the thing is, if the birds reach the island and they get there and there's still a full snow cover, then most of the animals will just eventually go elsewhere and not breed on that particular year. And so there are some years in the record in the last 30 years where you have less than 10% or 20% of the animals that do breed on the island simply because the snow is too abundant on the island. And so those have a fairly strong reaction to the spring conditions that you do find on the island, and presumably with the warming, eventually the snow cover will melt or the snow cover will disappear earlier, which will facilitate their reproduction. There are other species that are also sensitive to the snow cover, but in different ways, such as the lemmings, who require particular snow conditions, allowing them to move up and down the snow column to get protection, but also to stay warm and access their food. And some years you have ice rain on the snow, preventing them to make those vertical movements. And in those years, we find high mortality and much lower reproduction for the lemmings. So they are also sensitive to temperature, but in a very different way. And then for all of the insectivorous bird species, each year, repeatedly, we have a peak in abundance of the arthropods somewhere around the middle of July. And if you have either a delay or an advancement of that peak in arthropod activity, this could have an impact as well on the breeding success of those animals because they require that high abundance of food to raise their chicks, and so the breeding success also depends on that timing of reproduction with the emergence of the arthropods. And lastly, but surely, some other players are coming from the south. So right now on Violet, you have Arctic fox who's dominating the carnivorous mammals. But there is at least one couple, and sometimes you have transient individuals that are coming to the island of red foxes that are not as well adapted to winter conditions. And what's critical for them, we see them right now on the pictures praying, but the advantage that Arctic fox has is that they are caching some food. So in the summer, they're just constantly attacking and picking lemmings and snow goose eggs, and they're making food caches which help them survive the winter. While the red foxes are not doing that, they're just feeding constantly. And so in the winter, it's a period of resource scarcity, and so they may not be able to cope with the harsh condition in the winter if they don't have the proper food availability. But with the ongoing climate change and different in the phenology of the other animals, this could eventually impact the performance of the red foxes. So all of this to say that there's quite a lot of variability in the way those animals do respond to the ongoing climate warming, and eventually got me to think more about this problem, so that we could eventually better anticipate what's going to go on with climate warming at the entire food web level. And so a couple of years ago, we started to investigate this notion of thermal mismatches. So we looked in particular at different studies who evaluated the response rate evaluated how different rates do respond to temperature. So we looked at attack rates, carrying capacity, and metabolic rate, fitting a traditional exponential curve. Sorry. And the activation energy is essentially the slope of that curve on a log scale. And we did found a very significant variability among the different species in the rate at which they do respond to temperature. And so in this study, we started to investigate a little further how this variability is impacting eventually traffic interactions, and more specifically, the amount of regulation you do have. So the amount of top-down control you do have by consumers on their resources. I was also inspired by another study that we did complete a couple of years ago. So I'm part of a research network that is called the Center for Northern Research, that do have a set of research stations along a south to north gradient from the boreal forest in Quebec up to the high Arctic. So the Violet Station is one of them, but they do even have a northern station on Alert, the military base on Ellesmere Island. And then there are also several stations in northern Quebec that do cover the transition from the boreal forest to the Arctic. And so we were part of a group of researchers that do have different expertise. Some were specialized on permafrost, other ones on wetlands, other ones on lakes and fresh water. There were obviously food web ecologists, but also vegetation plant ecologists, and so we did a big synthesis trying to understand how the entire ecosystem will eventually respond to climate warming. So, for instance, in our group, who were interested in invertebrates and the food web, we were able to identify how the different food webs vary from the boreal to the Arctic community. And then we tried to guess at how they will respond to climate change. And some of those players do respond very rapidly, either because they're highly sensitive to temperature, like the ones that I presented earlier, or either because they have fast demography, or because they have high dispersal capabilities. Some other species are highly sensitive to vegetation, and this was basically the key message we made. The key observation we made from the system is that the dependence on vegetation is key in the system, and vegetation is going to react much more slowly or much more slower than every other player in the food web. And so even though we have actors here that may respond very rapidly, at the end, the entire community will shift north at the rate that vegetation is moving up north, which led us to eventually develop some kind of intuition on or some kind of concept of burden communities. So the ones that are just basically slowing down the entire ecosystem. So typically, when we're looking at the resilience literature, uh, people make the assumption that you have initial state and eventually resulting state, and the slope of um, this slope here that you do have for the plane is telling us how fast the system is responding. And usually in the literature, people are making the implicit assumption that all of the components are reacting at the same rate. But the reality is that some are reacting much faster and other ones much slower. Like I said, vegetation is responding much slower than passerine birds, for instance, that do reproduce very rapidly and also disperse quite a lot. And if all of the components are independent, then each of them could eventually move at their own rate, and we're gonna eventually witness changes in the ecosystem structure. But if the entire ecosystem is tied together, like we believe it is in a network of trophic interactions, then you may eventually have burdens that will just slow down the movement, the response of the entire system instead of having independent responses. So that was an intuitive interpretation of what was going on, but so far we don't have theory to try to explain and better anticipate what's going to go with those changes. And it's basically the question that I'm asking: how could we anticipate the complex response of those ecosystems, given that some are responding much faster and some are much more sensitive to temperature changes than other ones? So I will just start with a simple cartoon. The idea is that you have an environment here, an initial environment that is eventually changing. And so you can imagine that it's also a time series here on the x-axis. And you do have a response, such as abundance of the different components of the network. And some species are responding positively. So like the ping ball here, it's moving up. Other ones are not changing much, and some are decreasing in response to that change in the environment. So we could eventually make a cartoon of the directions they're changing and the strength of their change. And we could do that for the entire network and compute the variability of those responses. Well, first, the mean, obviously. And so for some systems, all components will react positively, which means you're going to have a positive response, such an increase in abundance of all the players. But more realistically, we could imagine that in any food web, some will benefit from that environmental change, and other ones will be harmed by that change, which will eventually generate variability. And so you may have different levels from one system to another in the amount of variability that you do have among the different players. I guess this is you're all familiar with this because it's part of the response diversity network, this underlying ID that we're trying to formalize here. And so I'll come with one core concept and a subtle refinement of this concept. So the core idea that I will present today is the notion of ecological coherence, which we characterize as the distribution of the different responses the species have to a change in the environment. And here I talk about responses in a very broad way. So it could either be intrinsic response, so how their fitness is changing to the environment. It could be their equilibrium abundance, or it could be their distribution. I mean, there are different ways that we could quantify that response. And I believe the concept is general enough so that it could encompass those different measures of response. And ecological network coherence is more specifically referring to the co-responses. So you can, if you start to represent a community with a network, you're looking at the relationship between the different species in the network. And all of those responses, interactions between the different species could also be investigated with co-responses or correlation, if you mean. I'll just be slightly more visual with this representation here. So the idea is that you have some kind of intrinsic response. So here we could imagine fitness to environment response curves for the different species. And if we're moving from the vertical line here on the left to the line on the right, you will find that in this case, all species do have a positive response, but some are like the ones in yellow here is responding more than the one in, I don't know, is that brown, pinky response. We could imagine the same thing for time series. Instead of being a fitness to environment relationship, it could be trends over time that we may look at. We could extract that. And given a diversity of responses like that, we could, for all pairs of species, compute the co-response. So measure how correlated each pair of species is. And we could compute from that co-response matrix the frequency distribution of the co-responses. So we could do it for all pairs of species, or we could do it with the application of a filter and looking more specifically at the co-responses of interacting species only. One question that I had for a long time, working on trophic biogeography, I've asked myself for a long time, are interacting species responding to the environment in a more similar way than the ones that are not interacting? So if this hypothesis is occurring in reality, then we could imagine that from the distribution of all co-responses, if we're filtering only on the species that do interact, we may have a shift to the right of among the interacting species. Is that clear so far? Any questions? So no answer, so I'll presume everything's fine, so I'll keep going. Now, combining all of this together, my intuition, or I could say my hypothesis, is that we may have eventually disruptions in ecosystem functioning if the coherence is very low. My intuition is that if all species from a network of interacting species are responding essentially in the same way, let's say that they all increase in abundance at the same time, well, we're less susceptible to find a loss of traffic regulation, extinction cascades, things like that, than the opposite, which would be some species at one extreme are benefiting a lot from the change in the environment, and other ones are harmed by that change in the environment. And so I'm making this hypothesis right now that disruptions are simply more susceptible to happen when network coherence is low. So we'll go through quickly a simple theoretical example first to explore this intuition, and then eventually I'll go through empirical examples where I do explore the amount of coherence we have in different systems. So to start with, we have a standard general Latka-Volterra model here. So X is the abundance of species I, and we're looking at the change in abundance over time for every species of a community as a function of its own intrinsic growth rate, which takes a positive value for the plants, the autotrophs, and negative value here for the heterotrophs. And then this growth rate is impacted by the total amount of interactions you have among the different species in the system. Oh, sorry, here it should be J and I in this equation. So the A coefficient here are the interaction coefficients among all pairs of species. So there's a simple solution to this system of equation telling us that the vector of equilibrium abundances X is the result of the product of the inverse of the interaction matrix. And so keep in mind that the inverse of the interaction matrix is a useful quantity that tells us the net effect between all pairs of a species, not just the direct effect, but also all of the indirect effects. For instance, the effect of a carnivore that on a plant through the cascade that it has on the herbivore, multiplied by so this matrix of indirect effects is multiplied by the vector of intrinsic growth rates at equilibrium. And what we'll do now is that we're going to consider that there's some environmental change that is influencing the intrinsic growth rate of every single species in this system. And so we have a change in the intrinsic growth rate. And so we're interested in the amount of change that this change in intrinsic growth rate will have, the impact that this change will have on the equilibrium abundances of the different species. And so there's a way that is not that complicated to eventually get at the expectation of that change in abundances for the different species. Simply, we consider that the change in the growth rate is a random variable, and then we could take the expectation for that solution, and it tells us that the expected change is dependent on the average response. So the product of the average interaction coefficient times the average change in response, plus some terms for the covariance between the interaction matrix and the intrinsic growth rates. So here, this covariance essentially quantifies the relationship between the interactions you do have and the rate at which species do respond. And so the covariance will be high if the most impactful species, the ones that do have the largest impact on the food web, are also the ones that do have the strongest response to the environment. Otherwise, the covariance will tend to be lower. There's no reason so far to expect that this term here will be different than zero. It obviously depends on the expectation we have for the overall amount of change you do have in the system. But if some species go up and other ones go down, well, we could imagine that the entire expectation here will stay pretty much the same, unless you do have some strong covariance in here. The other result, and to me, it's the more important, the most important one, because it relates to the intuition that I developed earlier. We could not only look at the expected amount of response that you're going to have from the different species, but most of all, we could look at the variance in this. And my intuition was about the variance much more than the expectation earlier. I was making the hypothesis that you're more susceptible to have ecological disruptions, so major change in functioning. So let's say extreme changes in abundances. If you're losing if you have low coherence. And the nice thing with this solution is here that you have a term exactly for coherence. So it's the covariance in the response of the different species. And this covariance is multiplied by the interaction coefficients of the different players in the community. And so basically what this equation is telling us is that if you do have a positive covariance, it's going to increase the variance if you have a match of signs of the net interactions. So in the case of competitive interactions or mutualistic interactions, a positive covariance will generate an increase in the total variance that you're going to have in the response. Or the alternative is if you do have a dominance of antagonistic interactions, like predator-prey interactions, plus and minus here that do combines with the negative covariance, then you're also going to have a higher variance in here. So it basically confirms the original expectation, which those are results that Dominique derived with the working group we were working on. Simulations were done for this simple Volterra model for one typical topology that you do have here. So we just fixed the topology. Here, the color coding corresponds to different types of interactions, from the very positive in green to the very negative in orange. And we varied the amount of covariance we have among the different species. Here is the variance for that each species is experiencing. So you do have two species that is experience much larger variance than the other ones. It's not surprising. It's either the plant at the bottom, which is susceptible to a lot of trophic regulation by the herbivores, or you do have at the top some predators that is, well, an omnivore at the top that is also accumulating the responses of the different species and is also subject to quite a lot of variability depending on the directions of the change of the underlying of the lower species. And as I expected, if you do diversify the amount of covariances that you do have among the different species, then you do also observe a larger variance in the response. So that's it for the. Yep.
Before you continue, may I just ask you to go to the previous slide to clarify something for
Yep.
my own curiosity? So I'm just wondering the letter K. So when you're going through a focal species I and you're looking at all the interaction coefficients between I and species J, and so what would be species K here? And another
yeah,
species?
that's a good question.
Yeah.
I thought about putting a small figure. Maybe I should do it. We're actually looking at a triplet here. So
Okay.
we have a focal species that is interacting with two other ones.
Yeah.
So imagine you have a plant and two consumers competing for that plant, or you can imagine the other way around. You have two prey species that are subject to apparent competition through another one. The target species is all in one case, in the first case is a plant, and in the other case the target will be the predator. And the covariance is among the net interaction coefficients we do have here are among the target and the other two players, and the covariance is among the two players. I just realized that it would have been much easier with an illustration of a motif, but
Yeah, yeah.
basically it's summarizing information and the response you could have for exploitative competition, apparent competition, a food chain, and a integral predation motif. So
So
all
if
of
you
the different
had
cases you can have for triplets.
so if you had more complex motifs with more than three species, would you have to expand this or not?
Oh, that's an excellent question. I have no clue how we could eventually expand that to larger levels. But keep in mind that here we have these are the coefficients of the inverse interaction matrix, which means that we also account for all other pathways that you do have in the food web. It's not just the direct interactions. These are the net interactions, and so we could have non-null interactions between two species that are separated by five interactions between them because it's an entire chain. The thing is that in most food webs, the longer is a chain of interaction, the more indirect interactions are, usually the weaker they are.
Okay, thank you.
And so I'll now move to a one first example, and I don't know if I'll have time to go through the second one. Maybe I'm gonna ask to you if you're patient enough to go through the second one. So I'll do the first one, and then we'll see how much time we have left. So we tried to apply this to different types of data. So we started with the response of the cod food web that we have in the Atlantic, and we had time series for an entire food web before, during, and after the collapse that was experienced in the 90s. Um, and what we did in this case is that instead of having the direct response to the environment, we looked at the trends that species were experiencing, and we were computing if they were having synchronized increase or decrease in abundance instead of having their exact response. Well, this is basically what I was explaining. So for every species, we extracted what's called alpha here. It's the trend, whether it's positive or negative. It's basically the rate of change in abundance. We cross it among all pairs of species, which allowed us to extract whether or not species were co-fluctuating positively or negatively. And then we looked at the different components of the distribution of those co-responses. The way we did it, we developed hierarchical GAMs. I'm not going to go through the details, but basically we have time series for every species, and we extracted an average trend over time for every species. This is a hierarchical model with species-specific coefficients allowing us to modify the direction that each species is experiencing. And then we also have latent variables that do describe other components of covariations among the different species, and you have the residual uncertainty at the end. And so there's quite a lot of variability in the system in the directions of the changes. This is just the frequency distribution of the rate of change across the entire time series and all of the species. And so we could see that some species do experience positive rate of change, other ones are experiencing negative ones, and it depends on where we are in the time series, because as you have seen earlier, there was a certain period of stability followed by a crash, followed by a recent increase in abundance. And this is reflected by this figure here, where you have the time series of the distribution of the rate of change across the different species. So there was a lot of variability in the 80s, just prior to the collapse of the food web. In the early 2000s, at the beginning of the increase in abundance, but in the period where densities were the lowest, the rates of change were almost of zero for all species, and then eventually it started to diversify again, and with two peaks of variability in the time series. And so basically this here is this figure at the bottom is simply the width of that frequency distribution you have here on the second panel. So we looked at the amount of variability we have in the correlations between species. We did it in two ways. So first, from the top to the bottom, you have the pre-collapse, the collapse, and the post-collapse period. And on the left, you have the full distribution of the pairwise correlations. And then on the right, you do have a filtered distribution by focusing only on the pairs of interacting species. And so, as I said earlier, there's quite a lot of variability if you look at what was happening, especially for pre-collapse. So there was a lot of variability, and consequently, some species were going up and other ones were going down at the same time, meaning that, yeah, we had the full range of correlations among the different species. But if you do look at the ones that are only interacting, then there's much more positive correlations among the interacting species. Things were a little more spread out during the collapse, although I must say that during the collapse, the abundances at some point got so low that it's much harder to evaluate with confidence the directions and consequently the correlations between the different species. And then at the post-collapse, we had a fairly different distributions of those correlation. To me, it's an indication that the system is still undergoing a reorganization right now. We are far from the conditions we had prior to collapse. The bonus with this is that we were able to revisit, and that was the original objective that we had with Catherine. We were able to revisit the well-known Living Planet Index. So we have a representation of the LPI here with the clear indication of the collapse with the negative trend that peaked in the middle of the 90s, and then the recovery phase. The LPI, we criticize it, and others did as well, for its inappropriate representation of the uncertainty. The novel way we do have using hierarchical GAMs better represent that uncertainty, and so you can see that there's quite a lot of variability at the beginning of the time series. If you do compare to the LPI, and the shape of the curve is also quite different from the LPI. The main advantage now is that we have a bonus, which is the measure of coherence, so the variability in the rates of change that could be interpreted at the community level. And it's a useful quantity, I believe, to better understand how ecosystems are reorganizing and the forces at play within the community are subject to changes. I see that time is flying. It's already 45 minutes, and so I will not go through the details of this example, but I'll just say that we basically apply the same reasoning to another type of interaction network, but instead in this case we use spatial data instead of temporal data. And so the idea here was to essentially capture a proxy of the fundamental niche, and we basically ask if the fundamental niche of the different species within that network matches also the interactions. So coming back to this original hypothesis that I had, that interacting species are more susceptible to share their environmental requirements. And it's basically for those hummingbirds and other interactors in a pollination network in the Atlantic rainforest. Here it's a mutualistic interaction network, and there's quite a strong evidence that interacting species are more similar in their response to the environment than non-interacting species. I'll just skip that. I could come back to it eventually if you do have other questions. So, in summary, I presented two concepts. Well, basically the one of ecological coherence with its variant, which is focusing on ecological networks. I presented it as a way to quantify the response at the network level to environmental changes and as a way to eventually anticipate if novel situations could lead to significant change in the functioning of those networks. I showed you a theoretical examples that makes the argument that we could have ecological surprises in situations where we do have a mismatch in the responses to the environment. That amount of coherence may also vary both over time and space, as we found in the two examples. And lastly, I haven't detailed it quantitatively, but all of those examples and the theoretical one and the time series and the spatial ones show that could eventually be applied to multiple types of data and types of problems that we may be interested. And I'll come back to the introduction where I talked about the system in the north. Uh, what I learned over time doing field work in that system, for this one in particular, it's not necessarily the case for all systems, but for this one in particular, ecological interactions do matter and do matter a lot. And so we definitely need to have a measure or some way to anticipate how the entire system will respond to environmental changes. Unfortunately, if you look at the network literature, well, people doing network ecology are used to investigate empirical networks, but the foundations, the key elements of network theory are not, to at least it's my opinion, they are not well tested empirically. Just to take one example, there's a lot of theory on extinction cascades, and I have never seen any single paper trying to test if those extinction cascades happen the way that we anticipate them doing network extinction experiments. Um, but now I believe that the concept that I presented is well rooted in empirical data analysis and could be quantified with available data. And so I believe that it's something that could be useful to both monitor and maybe eventually anticipate how those communities are changing. I'm just putting that in perspective with the ongoing explosion that in biodiversity metrics that we do have with the global biodiversity framework that was made a couple of years ago. There's been a diversification of diversity metrics that is just incredible. There was a review last year that shown that currently there are more than 500 different ways to monitor biodiversity changes. They look at what they're actually changing. So monitoring, is it about the state of the system, the pressures, the response, the benefits, and things like that. We do have metrics that are applied at the genetic level, some at the species level, some others at the ecosystem level. But as far as I know, there's not much of them that do look at how communities do respond. And there's not much of them that help us anticipate how they will respond in the future. And so, well, we're going to keep going with this measure of ecological coherence with the hope that eventually it could be a useful tool to monitor biodiversity changes. With this, I'm done with my presentation and I'm ready to take questions.
Thank you so much, Dominique. Yeah, if anybody has questions, please unmute yourselves or ask your question in the chat. I know I have a gazillion questions, but I'll let others speak first. I guess I can ask my question if nobody wants to jump in. I have, as I said, I have so many questions, but the first one that's I think I'd really like to hear you talk about a little bit is that from your cod food web example, it seemed to me that it kind of refused your earlier hypothesis in that if species respond in the same way, that we expect fewer cascading extinctions. So if your ecological network coherence is high, then you have less of a collapse. And it looked like when you showed your graph, it seemed that you had high variability, but some ends pre-collapse had very high values of ecological network coherence. So I'm seeing a lot of things there. Part of it, it looks like there's some hysteresis sort of behavior going there from like the early warning signal sort of literature concepts. But could it be that it kind of refutes your hypothesis in that if all of the species are responding in the same way, which is what response diversity theory would tell us, like there's low response diversity, then maybe that caused the collapse. So you said, if all respond positively to climate change, then the network is fine. But you could also see the opposite, right? If all are responding negatively and your system is closed, then you don't have any room to adapt. So do you want to speak a bit to that?
Yeah, that's an excellent question, and in honesty, I'm working with Catherine right now to finish the paper, and I basically had the same question to her recently when I visited her. I realized that in our paper we were able to document how the response do vary over time. We looked even further at the co-responses. We found a very significant change over time, but we had difficulties in interpreting in relating those observations to the actual collapse of the food web. And when I came with her, but I don't have the figure right now, we had the idea of making a plot where we could represent the average and the variance, and you have locations in this plot where things could go well. If the average is positive and you have low variance, well, everyone is happy and things are fine. If the average is negative for all players, you have very low variance. Obviously, everything is going bad as well. Imagine that. That this actually may be one thing happening in the cod example. If you do overfish all species, you're going to have a negative trend for all species with very low variance, and the whole thing will collapse. But then the notion of coherence, I believe, comes somewhere in the middle. It's not because you have, and this is a problem with the LPI, for instance. You can track the average change of a bunch of species in a system, and if the average is telling you that overall nothing is changing because some are going up and other ones are going down, well, you may have the wrong interpretation that everything's fine and everyone's happy because they're just at steady level. They could indeed be a steady level, and so in which case you're going to have low variability among the different species, and they will all be at zero. But if the average is zero, but it masks a huge variance, that means that some are just taking over and other ones are crashing, then there's something wrong. And so you're just pointing to a big gap that I have both in my presentation and in the paper. We need to have that kind of a summary way of interpreting the directions of the change and where to be concerned and not be concerned.
Yeah, I would maybe have a look at the early warning signals literature because they have some neat metrics to look at variance trends and hysteresis. And I was just wondering if this indicator that you developed could be used as such. But yeah, it'd have to be tested.
Personally, I just enjoy and I love the early warning literature. I find the concept super appealing. my only concern is that it does apply to some contexts and not always.
Yeah,
And well, you don't necessarily need to have hysteresis, but usually you need to have other conditions for it to apply. And so it's not a general concept. It applies to some systems and not others. And so I find it a bit risky and too specific to stick with this. But it's nonetheless a useful thing to at least try to relate this concept to the early warning signs. Um, there is some we could find and derive from the literature some predictions about the covariances in the early warning signals. It's just that it's never been very explicit, but the theory is telling us that prior to collapse or prior to major change, you should have an increase in variances. Well, if we look at some papers, they're also telling us that we should have also a change in the covariance structure
yeah,
prior to collapse. But this hasn't been, to my knowledge, made very explicit because there's no multivariate version of the
yeah,
early warning sign literature.
yeah. I guess we're almost at time. Does anybody else have a pressing question they'll want to ask quickly? I guess not. Thank you so much, Dominique, for being available to do this presentation today. I hope everybody enjoyed it. I certainly did, and I probably will email you with a bunch more questions.
Pleasure,
Um,
pleasure. Thanks for the invitation. It was really a pleasure, and it was a good, kick in the butt for me to assemble that presentation. I haven't made it before, and so it was good to refresh. It's easy over time to give always the same seminars, and so I had this opportunity to just create a new one from scratch, and that was a good thing. It was a good way as well to value contributions of the different students. And I should say, I acknowledge contributions of Catherine and Alex, but they are not the only ones. As I said earlier, Dominique contributed to some of this work, and there were other students involved as well. So thanks to them.
Thank you, Dominique. I'll let you go. Bye-bye.
Well,
Bye,
bye
everybody.
bye.
Thank you.